It's okay, sound? I'm glad to hear you talk back to me, so we're already interacting. That's fantastic. I love to come to something that's called Creative Fridays because I think about being creative every single day. My life is based on creating things. And, you know, a lot of times it's harder than you think. And we live in a world where people have demands and deadlines and, you know, they expect things right away. And people say, well, you know, when are you going to get this done? And I go, I don't know. No one's ever created this before. And it's going to take some time. You know, we have to try things. And so that's the frustrating thing. You know, sometimes I go, what do you think of this idea? And they go, well, it's interesting. Why don't you develop it a little longer? Tell me where you've gotten with it in about 30 years, you know. I mean, creativity is a struggle. And, you know, but the reward is that we can create things that have lasting value. You can have an idea you're the first person that created that idea that could last basically for the rest of the history of mankind. You could produce an image or write something, you know, that'll last as long as civilization does. And the satisfaction, even though it's a struggle, and sometimes we don't get the monetary rewards and the things right away, that it's worth it because in the long term, you can have the satisfaction that you can make a true difference that lasts, you know, for generations even. So I am interested in how the brain works. And I'd like to know how the brain works in order to be able to use that knowledge in order to treat blindness, to give vision back to blind people. And, you know, if you're going to try to do something like that, you have to start somewhere. And kind of like with a test bed of some sort. And I'm so thrilled to be here, something called color, because I love color. I think of 16 hours a day, I'm thinking about color. And it turns out that about 1 out of every 12 men in the United States has some form of red-green color blindness. So it's the most common genetic disorder in people. And these people, you know, they're color blind. So they have a form of loss of vision. And there are monkeys that are like people in that there are some individuals in the population that have normal color vision, but other males, like this guy right here, his name is Sam, was born with red-green color blindness. And we've developed a method of curing color blindness. We've been able to cure color blindness in Sam. And, but in order to really kind of, this opens a whole idea basically about how our vision works and how our brain works, and gives us things that wouldn't be possible to think about, you know, before we were able to do this kind of thing. Well, as a scientist, one of the things that we need to be able to do is we have to understand the relationship between the visual stimulus and ultimately that thing inside of our head, our perception. And so we do that as scientists, but the first people that did exactly that kind of thing were not scientists, but rather that this has been the occupation for, you know, a very long time of artists. They're studying, people that are artists study, you know, basically what can I produce on campus or in a sculpture that ultimately produce a particular percept inside your head. And that what artists realized is really, I mean, we have this idea that we look at something and somehow we're just creating an accurate representation of that thing inside of our head. but it's not really true. That the physical world out there is really quite a bit different from what goes on inside of our head. And one of those things is the physical stimulus for color. Probably many of you know that color, the physical stimulus, is something about the wavelength of light. And, you know, the visible spectrum goes from 400 to 700 nanometers. So, like, if you break it into little 1 nanometer increments, there's maybe 300 different wavelengths. And those can occur, though. You can get things that reflect wavelengths at all different combinations. And so there are literally an infinite number of different things that could be colors. But we don't see an infinite number of different things. And Leonardo da Vinci was somebody who first wrote something about this down. And he said that there were not infinite number of colors, but he said that there are only 6. And he wanted to put them in order. For what reason, I don't know. The first one is white. Even if some philosophers accept neither white or black in the range of colors, and we all kind of recognize that black and white are different than other colors, and we actually use a different word. We use the word hue for things that are red, green, blue, and yellow, where, you know, black and white isn't one of the hues. Hence, white is the first of the simple colors. Yellow is the second, green is the third, blue is the fourth, red is the fifth, and black, for whatever reason, is the sixth. But amazingly, there's a very different thing inside of our head, just 6 colors, even though there's an infinite number of different combinations of wavelength in the real world. Well, of course, Da Vinci was able to use color, this kind of basic, you know, colors, you know, to great effect. And you can see here that, you know, like, you know, there's red, green and red and blue and yellow and black and white, the 6 colors. You know, but the thing that happened is that, you know, really when you look, that what's up there, this is just plaster and paint, right? But it's magical. The magic is not, though, in the paint and plaster. The magic is in the ability of the artist to understand the relationship what's up here to ultimately what you're going to create inside of your own head, you know, to make something that's so beautiful and also elicits such a great, strong emotion in us. Well, this whole principle about how contrasting and beautiful the 6 basic colors are something that's been used in art, you know, throughout the ages. So here we see red and green, blue and yellow, black and white. You know, and here's a Van Gogh. And it's not just one culture, but you can see the same kind of use of these fundamental colors across cultures. So as I said, you know, there's an infinite number of wavelengths, and those exist in an infinite number of combinations. But, you know, that's not what we see. Now, people that, you know, are involved in any kind of art know one thing is that you can use 3 primary colors to mix and get pretty much all the other colors in the world. And all of a sudden, I'm not saying that there's, you know, 3 basic color sensations, but there are 6. You maybe also know that human beings have 3 different kinds of receptors in our eye. That one most sensitive to blue, one most sensitive to green, and one most sensitive to red. And the fact that a video display like this only uses 3 different colors to make all the colors we see is not because of anything about the physical world. It's because we have 3 kinds of receptors in our eye. If we had 2 kinds of receptors in our eye, we wouldn't need to have video that had 3 different colors. 2 would suffice. But if we had 4 receptors in our eye, then we'd have to remake all of our displays and cameras to have 4 different receptors. And so, so we kind of have this idea that 3 is an important number in color vision. It is because we have 3 different kinds of cones. But actually, we have 4 hue sensations, red and green, blue and yellow, and then we have black and white. So you have 3 receptors, but you have 6 sensations. Why is that? Well, if you have a digital camera, this is the array of pixels in a digital camera, and there's 3 types, red and green and blue. And at the back of our eye, we have cells called cones that are responsible for us seeing color. And this is a cartoon of me drawing what it looks like end on when you look at the cone photoreceptors in your eye. And this would be what it's like very close to where our central vision is. And there's 3 different kinds of receptors. There's red and green and blue. Now, we're a little bit different from the video camera. One thing, we're a piece of biology, so the red and green cones are kind of randomly intermixed, and our brain doesn't, but that doesn't bother it. And there's about 5% of the total are blue cones. Again, that seems a little odd, but our brain is able to extract information about blueness with having a small number of blue cones compared to red and green cones. Now, if we had a sensor that only had 2, had one kind of detector, we actually get 2 colors from that, because the detector can be at some high level, and then we could say it's white, and low level we say it's black. So actually, from one sensor, we always can get kind of 2 sensations, either when the sensor is on or when it's off. And our vision, and this is why we have 3 receptors, but we have 6 sensations, because from every receptor, we get 2 sensations. So, and I'm not going to show too much biology, but these cone cells, the photoreceptors, are kind of long, skinny cells, and we were looking down the end of them. And they're kind of closely packed. And so this is what a cone cell kind of looks like. But it turns out that every cone cell actually sends 2 signals out of your eye to the brain. And it sends, and what it's doing is it actually has a network that each cone is able to get information from the surrounding cones. And what happens is that it compares the amount of light it's receiving from the amount the surrounding cones are receiving. And if it's receiving more light than the surrounding cones, it sends a signal out one pathway, and it says, I'm seeing something that's white. And if it's getting less light than everything that's around, it says dark. So you actually have 2 kind of labeled lines going out of your eye. The one says light, dark. Okay? So with one receptor, we actually have 2 signals. We have an active signal that says blackness and one for whiteness. So what happens when people are red-green colorblind is that they're missing the gene for either the red cones or the green cones. Now, luckily, that it turns out when the gene is gone for the red photopigment, all the cones that would have been red, they just express the green photopigment. And so the sensor array for people red-green colorblind is completely normal, except for instead of having 3 different kinds of cones, they only have 2 kinds of cones. And what happens in order to give us color vision, that your brain compares the output of the blue cones to the green cones, and it gives you 2 different color signals. And so you, in places where there are no blue cones, you get kind of a black and white signal, and where the blue cones are, there's a comparison made between blue cones and green cones that gives you a second color sensation. So you get 4 sensations with just 2 kinds of cones. And this is what the rainbow might look like to somebody who's red-green colorblind, because those 2 sensations, when you stimulate the blue cones, you see blue, and you stimulate the longer wavelength cones, the red ones or green ones, then you see another color. I like to say it's yellow. And so that's your world. So if you have, here's the big tray of Crayola crayons. And there's about 104 crayons or something in there. But you see, if you had no color vision, they're all shades of gray. But for people that are red-green colorblind, they have black and white sensations, and then they have two other sensations that I'm calling blue and yellow. Now, it turns out if you're a little colorblind boy, you do not need 104 crayons. You only need, well, you know, it would be nice to have 3 crayons. You can have a blue crayon, a yellow crayon, and, you know, just so you can make some black lines on the white page, you want a black crayon, right? And so the amazing thing is that with just 2 sensors, you miss out on a lot of the colors. And what happens then is when you add a third sensor, you actually add 2 new sensations. And those 2 new sensations we call red and green. And those come about from comparison of the red and green cones. So, and then what happens though, there's something magical about adding a third sensor. Because what happens is you have new sensations, so there's a possibility of adding a little bit of redness or greenness to all the other colors that we see here. So, you know, if it's a white color, we can add a little redness and we get pink. And if we don't have very much whiteness, you get a pure red. But if you add, you take a blue color and you add some redness to it, you get purple. And if you have a blue color and you add greenness to it, you get aqua. And so what happens is that, you know, you now had only, you know, basically black and white in 2 colors, but now you can add these 2 new sensations to all the other colors, and you go from needing 2 crayons to all of a sudden you need 104 crayons, or maybe even more. So there's something fantastic about that. And color is fantastic, you know, and you can see the huge difference between, you know, living in a world that you're red-green colorblind and what, you know, people live in that has normal color vision. But what I want to get to is, you know, people, if we all had 2 cones and I told you, oh, I could add an extra cone to your eye, and you would see a whole entire world that you've never seen before and be totally different, and it would be like amazing, you would go, oh, no, that's not true. That doesn't sound right to me. Because as you look around, if you're red-green colorblind, you look around and you go, I honestly believe that what's inside my head reflects what's out there in the real world. And so if I'm not seeing it out there, you can't tell me just adding something new to my head is going to now, you know, change the whole entire world around me. Because you don't realize it's all constructed right here. So what I would argue is if we added a 4th kind of cone, we would have access to a whole entire world of spectral differences around us that were lost on us. And then, you know, we could be, you know, the person that had the extra cone could be, you know, we'd add that extra color, and now we'd have to have 1,000 crayons or 1,000,000 crayons. And you go, oh, look at all those colors. But it's so amazing because we don't think that this is the way that we work. You think that you're kind of gathering all the real world out there, but it's a little tiny sampling of the real world that's being created for our sensations. Well, so I'm interested in the neural physiology of perception. And, you know, these are kinds of some of the things I think about. But ultimately, you know, the possibility of using some of that knowledge in order to better the human condition, and in particular, do things like cure blindness. Now, so what is the potential for restoring sight to the blind? As I said, you know, we have to start somewhere, and that one thing, I wanted to do something where we had an animal that had a loss of vision that was like what humans have, but it was a little simpler problem than somebody that was completely blind. Now, it turns out that in the animal kingdom, animals that are blind don't survive in the wild, so there aren't blind animals in general that, are animal models. But there is this monkey species that lives in South America that has red-green color blindness like we do. And as I said, you probably all know that dogs are colorblind. They're actually not completely colorblind. They're just red-green colorblind like many mammals are. And that's a little different because all dogs are that way. But in these monkeys, that some of the individuals, just like humans, have trichromatic full color vision, and other ones are red-green colorblind. And so we wanted to be able to take some of the colorblind ones and use those as a model to see if we could treat their vision disorder. Now, these particular monkeys are spider monkeys, and portrayed in this beautiful picture from Frida Kahlo. And the monkeys that we work with are closely related species. They're the squirrel monkeys. And you look at this picture and you go, I don't think that's really true, that the monkeys come and hug you and they're so sweet like that they are with Frida here. But it is fact true. So these are not my monkeys. These are not my monkeys in the laboratory because we had to take precautions that we don't give them diseases that we might pick up. So we have to be careful about our contact with them. But this picture was taken at a resort in the Amazon. For years, since the 1970s, they've been leaving out food, and these monkeys come to get fed. And when you're walking along, so you can see there's a monkey right up there. Walking along, and all of a sudden you feel something jump on your back if you're wearing a backpack, and the monkey comes and unzips your backpack and reaches around in there to see if you have anything good to eat. But anyway, these are these wonderful, docile, beautiful animals. So what my argument is that the way that our eye works is that there are certain regions of the retina that's like, just like I said, there's only one sensor where you'd have black and white. Because there's only one type of cone in certain little regions where comparisons are being made. And that those little pixels are the ones that are giving us the senses of black and white. And the other parts of the retina that are nearby that are analyzing the same part of the visual scene where there's a blue cone, those are what we use for getting the sensations of blue and yellow. And so you have these different lines that are analyzing this little region of space, and you get black and white and blue and yellow. And what I want to argue is that when you have three different kinds of cones, what happens now is that now this place right here that always was a green cone has a possibility that was talking to the blue cones, has the possibility of also could have a green cone in it next to a blue cone. And so now you get 6 signals that come out of your eye, black and white, one's for blue and yellow, and one's for red and green. And so the idea is that in an adult monkey that's lived all of his life as being colorblind, if we could put a cone mosaic into their eye that looks like this, it would transform a subset of those cells that were transmitting information about blue and yellow into red and green and give them the same kind of signals going to the brain that a normal trichromat does. And so that was my idea. And that what we could do is we could take a gene for a red photopigment, and we selected animals that were the squirrel monkeys that have blue and green cones, but no red. And then we took a human red photopigment gene, and we packaged it into a vector. that was originally derived from a virus that has ability to penetrate cells and inject DNA and turn that gene on. But that if we can adjust kind of how much virus we put in the eye, there's not enough to infect all the cones, and it's kind of random, that we could produce a random mosaic in the eye that basically looks just like a normal retina looks like. So this whole concept of injecting a gene into the eye, it's gene therapy. And the way that we did it is that we have this solution containing this viral vector, has the red photopigment, injected in the eye, and it turns out the way the eye is, I know, it's only one little shot. This will hardly hurt at all. The animals, they were under anesthesia. The photoreceptors are actually behind the main part of the retina. So in order to get the solution to the photoreceptors, we actually had to put it behind the retina. It actually makes a little detachment, and we made a couple of different injections to make sure that we kind of covered the whole back of the eye with the solution. And as I say, it was adjusted so that it would infect a subset of the cones and produce a random mosaic of new photoreceptors. Now, what's happening is that we are infecting the green cones with the gene that expresses a red photopigment. And what we're trying to do is turn the green cones into red cones. And and happily, this new gene we put in, the green cones say, I love this new gene. I have good access to it. I can turn it on. So well, in fact, that it actually outcompetes the green photopigment that was already there. And even though there's still a little green photopigment expressed, that the cones do become mostly, that get infected, become mostly red cones. Now, well, it's not a question of whether or not we can inject something into the eye, infect it with the virus, and turn on a new gene. We could demonstrate we do that, but that's not the question. The question is, does this change the way that the animals see in a meaningful way? And so what we wanted to do is we wanted to do a color vision test. And probably most of you had your color vision tested. And the most common thing is there's a book, and you open the page of the book, and there's kind of a bunch of dots in there, and there's a symbol, like a number or a letter, something like that, and against a background of a different color dots. And if you're colorblind and you can't see the difference in color between the symbol and the background, you can't say what's there. But if you have color vision, you can use it in order to get the right answers. And we did the same thing, and it turns out that there's a commercially available computerized version of something like that. It's called the Cambridge Color Test, actually developed at Cambridge University. And we talked to them about our idea, and we said, well, it's hard to get the monkeys to say, oh, I see the number 12. And so what we did is we modified the computer program. So instead of being a number or a letter, it's just kind of a blob of color against the background. But for the monkeys, what we did is we could move the blob of color around. And we could also change the amount of color that's added to the positive little stimulus. And so we can make it closer and closer to gray, so we can actually measure their threshold to see how acute their color vision is. And so the task then is that the monkey, it's exactly like testing a little kid for color blindness. The little kid, you explain to him, what I'm going to do is I'm going to show you a page, and I'm going to ask you to tell me what symbol you see. And we flashed this thing up on the screen, and the monkey has learned that when we do that, he's supposed to tell us where the blob is, and it occurs in one of those three places, and he should just touch. All he has to do, he said, okay, I get it. Now, I can tell you what I see by touching the place where I see the blob. Now, okay, so these monkeys were treated with this gene therapy, but we wanted to test their color vision before we started, and we had to train this in this color vision task. And you might say, wait a minute, you have colorblind monkeys and you're trying to teach them to do a color vision task. But it's no problem because they have color vision for blues and yellows. And so we trained them to pick out the blue and yellow things, and then we could sneak in the red and green ones in order to test their red-green color vision. Interesting that because the way our brain analyzes color information, that if you're red-green colorblind, this blue I'm talking about is not exactly the same as our blue. And the yellow is not the same as our yellow. And the very best yellow actually looks kind of greenish to a colorblind person. Or the best yellow to a colorblind person looks greenish to us. And so that so the very brightest, most beautiful yellow looks a little green. And what I'm showing you here is the monkey taking this test, and the stimuli are this yellow stimulus, but it looks green. And in the video, it even looks a little greener than it looks in real life. But here's the monkey showing you what great blue-yellow color vision he has. Okay. I had the, unfortunately, my sound, I checked it beforehand. All right. The sound is worth it. Okay, but I may have to be creative here.
The volume on the side looks like it's off. Can you see the little, yeah, on the, make the bar show up again? And then yeah, you look through the little icon? Oh, it's up already, okay. Start playing.
Okay. Why, thank you. The day is saved. You don't have to hear me imitating the sounds. Okay. I told you that he touched the window, to say where he sees it. But what I didn't tell you is that he actually touches it with he's actually touching it with the tip of his tongue. And we don't care. I didn't say how he had to touch it. Just he had to touch it. And but every time he gets it correct, there's a little drop of grape juice that appears in a well directly underneath where he gets it right. And they like to get lots of grape juice, and they figured out that the fastest way to get things to work is that they kind of touch with their nose and then down and get their grape juice. And that's much better. Touch, get your hand out of the way, and so. And the other thing is that just like when you have the kid, and you get testing their color vision, and they get it right, you say, "Good job, that's right." And so that every time they get it correct, they have a little clicker, a clicking sound. And that click, as soon as they hear that sound, that tells them that they got it right. And that the juice will appear. And so that kind of serves as a secondary reinforcer that just hearing the sound, they go, "Oh, I got it right. Everything's going extremely well." So I can make a display to show you exactly what the world looks like to a red-green colorblind monkey. It turns out, unfortunately, I can't make a display that shows you what the world looks like to a trichromat to somebody who's red-green colorblind. And so here's this image right here of the monkey looking, and that's the positive stimulus, but there is a red and a green color that he cannot tell from gray. So again, it's amazing, but you think you lose your red cones and you just lose the sense of red, but that's not the way it works. You don't have any sense of red or green, and so those things look exactly gray, or there is a reddish color that looks exactly gray. So it looks like this to us, but this is what it looks like to the colorblind monkey. And this is actually what a monkey looks like to a colorblind monkey. So they're trained. Actually, they get up and take a color vision test every day. And we've been doing this for years. And they're trained when it comes up, here's the thing, where's the colored blob that they know we're asking? And all of a sudden they're looking at this and they go, I don't see it. And this is very frustrating to them. And so normally the way we do it is we put lots of blue ones and yellow ones in there, and then we sneak in some red ones and green ones. And that makes them so they don't get too frustrated. And that way we're able to test their color vision and show just exactly how good, and in this case bad, their red-green color vision is. But just for your entertainment, I have a sequence here. Well, we didn't give him any blue ones or yellow ones, just the ones he can't see. And so this is, a demonstration that the monkeys are truly oh, wait, sorry. I've gotten it. Here we go. So there's this nice red blob there. Yep. All right. Okay. So just like we have to tell him that he got it right, we tell him when he gets it wrong. So when he gets it wrong, he gets a buzzer. And then he gets a little time out. We're not going to show you another one for a minute, Just think about it here. And of course, occasionally he's going to get it right just by guessing. And a lot of times if he guesses in a particular spot, he'll go right back to that same place again. What? Okay. So, scientists are skeptical, and we knew that if this would work, that people would say, oh, well, monkeys are smart, and maybe your color vision test isn't that great, and, that over a period of time you gave them some treatment, but their color vision didn't get any better. It's just that they figured out some trick to get around your test. And so we actually ran these monkeys every day for 2 years before we treated them. And their color vision, they learned the tax, and we were able to get a measurement of their color vision thresholds, and then we watched them over a period of 2 years, just to make sure that no one would say, no. And very soon after we made the treatment, the animals began to see reds and greens that they'd never seen before. And it's really quite remarkable. And it was one of those things I go, wow, it was actually a long, frustrating experience. And you can see, it sounds like a long time. 2 years we waited before we even did anything. But actually the process was much longer because we had lots of frustrations and things didn't work right. And, we developed, this color vision test I'm showing you and how to train the animals. There was a lot to it. But, ultimately, the monkeys did see red and green that they hadn't seen before. Okay. And so here it is. Here's the monkey. This is actually about a half a year after he was treated. Okay. And now you see every once in a while he's not so accurate, so he misses, but you can see that he's able to look. And we can make the amount of red in the stimulus less and less and less. And you'll see that some of these, there's not that much red. We get closer and closer to threshold, and yet the monkey still sees it. That is good. Yeah, it's amazing. Okay. But what I want to argue, so when we did this, we were so like, wow, that is so fantastic. But I want to argue is that what happens is that right away we gave him a new sensitivity, and he was able to, soon after that the gene got turned on, when it's up against a gray background, he could now see that blob we hadn't seen before. But I said that really what I think happened is that he had these red and green kind of channels that go up to his brain, and we converted some subset of the red ones and green ones into, had blue and yellow channels that went to the brain. We converted some of those into red and green ones. And so I think really probably what happened is that this new thing came up, and it made a little buzzing play somewhere in his brain. He said, oh, that's blue, and he gets it right. But he didn't actually see red and green. That's what I think. And people, but nonetheless, right away that we cured them of their color blindness in that they could take this particular color vision test, and they could see something they had never seen before. So it's encouraging that ultimately the same kind of technology could be used for other kinds of blinding disorders. So now to get to some of the questions, like as David said, all the time that I was a little kid, I don't know how many times somebody said to me, wait a minute, what if my blue is like your red? Like I'm wired up, the opposite. So when I'm seeing, when I'm looking at a blue thing, I see the same thing as you see as when you see red. And we all went, ooh, that's cool. But that's not the way our brain works. I'm saying that there's no genetically encoded labeled line that goes up to your brain that says red or green. That there's neurons that fire action potentials, and they go to some place in your brain, and their action potentials occur at different times depending on what colors you see. And what happens is that over a period of your lifetime, you will learn that that particular vibration in your head is associated with particular things in the outside world. So you'll learn that some neuron up here is buzzing, and I'm looking at some bright red lipstick. You associate that, that particular sensation with, bright red lips or watermelon or stop signs. And that what I want to say is that we do all live in our old kind of worlds where we see a color because that color is something that you say red, but it's really an association with all the things in the world that we've experienced that are red. And when we look at red things, it vibrates those particular neurons. And when we look at other neurons, we vibrate, when we look at greens, we vibrate other neurons. And we've learned that those are something else. And when I look at green, I see pistachio ice cream. And I associate green with grass and algae, green leaves, creme de menthe. And so it's not like we're pre-wired with some kind of, thing where you look at a red thing. You actually have to build this experience in. And, we're basically looking around and experiencing color that is something that we've learned and associated over a whole entire lifetime. And this is part of the reason we love color so much, because you all of a sudden look at a particular color and it can call up all these very strong kind of emotions that are associated with all the other kinds of things. Red, blood, And so that's my answer to the question of, whether my red is the same as your red. And what I believe, so this didn't occur to me at all when we first, were treating these animals, or what's going on inside your head. I didn't think I had permission to think about this because, it's kind of all pie in the sky. You can't really do an experiment. You can't, but then we did this fantastic experiment. And so what we began to do then, I said, well, okay, if that's true, that the animals wouldn't be able to tell, maybe they can't tell blue from red, even though they can tell red from gray now. But it took me a kind of while to get up to this idea. And it turns out it's really easy to ask them any color question you want. You can ask them if they can tell blue from red. You just make the background dots blue, and the thing that they have to find is red. And if they can tell the difference, then they can pick the red things out in the blue background. And over a period of time, I tell people about this. Oh, look, they can tell blue from red. And they say, can they tell red from green? Can they tell yellow from blue? I go, I'll go find out. And so I, went and we tried every possible color. And it turns out that, now, the monkeys, this is actually probably a year by the time we got, all the different colors tested, but over a long period of time. And we're in there every day with M&Ms in our hand, holding them up. What do you see? Can you pick out the red ones? So we gave them a lot of kind of color stimuli, not really scientifically, but just because we were kind of curious, if they'd react to different kinds of things that were different colors. And so we gave them a lot of things to associate with the different colors. And ultimately, at least, what we can ask them what they can see, they're ultimately able to see all the colors that, a normal trichromat sees. And so I'd like to do this in a person so we could talk to them about it. But, as far as I can tell, and what we can tell from looking at monkeys, this is what we did to them. But I don't think, I actually was caught on videotape saying, oh, it must have been so great because one day they woke up, they opened their eyes, oh my God, it's Technicolor, but I don't think that that's what happened. I think they woke up one day and the world looked just exactly the same. But they went into the box and they got it right every time. And it was like, hey, this is pretty good. But over a period of time, those lines inside of our head that, buzzed were different when a blue thing was there than a red thing. And different when a yellow thing compared to the green thing. And that ultimately, they learned to do this. And I think it speaks to the capacity for all of us. This is part of the reason why the first time you ever, maybe drank a glass of red wine, somebody said, oh, this is a good one, this is a bad one. And you go, oh, they both taste like vinegar to me. But over a period of time, we learn to associate, all the different subtleties. And I think that this is kind of what happens in our head. And it's really what makes color so fascinating to us because it's tied, to all the fantastic things that we love in the world, or hate for that matter, or are afraid of. So, this is a scene that is been altered so that you get to see what a colorblind person sees. And I get emails from colorblind people all the time, and he said, my wife says, let's go out and look at the sunset. And he said, but, I go out and kind of looks the same as it does when it's not setting. And so here's a picture of a sunset to a colorblind person. But here's the same scene if you have red-green color vision. And here's another one. I get a lot. This is actually the Tokyo subway. And if you're in Tokyo, they go, how do I get, to so-and-so? They say, oh, take the magenta line. The colorblind person goes, But if you have full color vision, it's really easy to pick out which one is the magenta one. So at least now what I begin to think about, I can put another photopigment in my eye and give myself a fourth kind of cone. And then just think of all the things that I could see. And, we don't think about it, as I said, because we kind of think we do see everything in the world, but we don't. Not even close. And it'll be possible. Then we need to make new cameras and new projectors, and we'll put fourth cones in all of our eyes. And, the world, just think how fantastic, how much we love color now. Well, at least David said I should give you guys something to talk about and think about. So now you have something to think about and talk about. And with that, I'll stop. And this is a picture of the beautiful University of Washington campus. And of course, there's red and green, blue and yellow, and black and white.